folks. So this is the third audio for our lab chapter 20 in the lab manual, protists. So we're using our Protozal handout as kind of our guide. In the first video we talked about the free-living protozoa, and then in the second video we talked about two intestinal protozoal parasites, Entamoeba histolytica and Giardia, and we also mentioned the bonus protozoal pathogen that can cause urogenital infections and is sexually transmitted, which is Trichomonas. In this video, I think I'm just going to focus on a arthropod-borne protozoal pathogen called Trypanosoma Cruzi. Trypanosoma Cruzi causes Chagas disease, named after the Brazilian physician who described the life cycle in the early 1900s. It's also referred to as American trypanosomiasis, for this is a disease found endemic in South America, Central America, and we do have cases here in the United States. One book said that we have around 200,000 cases in the U.S. Another book referred to between 8 and 16 million cases, and again, this would be primarily in the Americas. Trypanosoma Cruzi is a vector-borne disease. It's an insect vector, a family called the reduvid bugs. They're often nicknamed kissing bugs or assassin bugs. And one specific genus of these reduvid bugs is the Triatoma insect, and hopefully we'll have some specimens in lab for you to look at, collected by Professor Dave Wyatt, our resident entomologist. Okay, so let's see if we can go find Trypanosoma Cruzi, the cause of American trypanosomiasis. It's a flagellate, and we'll see it has another cousin called Trypanosoma brucei that causes African trypanosomiasis. But again, this video, guys, will just focus on Trypanosoma Cruzi. The prepared slides are thick blood smears, and the reason you have to make this smear thick is often there aren't that many parasites in the blood. A lot of your slide boxes, the slides aren't very good. It's really, really hard to see the Trypanosoma Cruzi. So we'll have a demo scope set up on bench 4. So again, the sample we're looking at would be a stained smear of blood. And here we can see the extracellular forms that that can be found in the blood. They can also invade and live within host cells in an intracellular environment. This one's kind of nice. What I look for in the blood smears is kind of this C-shaped here. We have this dense staining little dot at one end. We can see it kind of comes around as a C. Here we would see the flagellum. It's the flagellum as it extends over the body of the cell. There's a membrane attached, so there's actually an undulating membrane here attached to the flagellum. So again, in the demo scopes, we'll be looking for these little C-shaped guys. So the natural cycle and transmission, as we said, this is a vector-borne disease, and it's kind of a fascinating life cycle. Excuse me. So these reduvid insects, they often are nocturnal, which means they come out at night to feed, for example, on humans. They might be attracted to the carbon dioxide we're breathing out because they love to climb up onto our face and bite near our mouth or near our eyes, and that might be one reason they're called kissing bugs. So they have a needle-like mouth part, so they insert it and they start sucking blood. And as they're sucking the blood, if the insects themselves are infected, they'll defecate, and it's in the feces the Trypanosoma Cruzi are located. So it's not that they're injecting the Trypanosoma in as they're biting us, they're defecating. And then we might be, sort of semi-awake and feel a tickle on our face, so we rub our face, so we end up rubbing the feces with the parasites into the bite wound, and that's how the parasite gets into our body. Or another way is that if we rub the feces into our eye, and this will often cause a swelling of the eye. This is called Romagna sign. And in endemic areas, this might be a sign that you have been infected with Trypanosoma Cruzi. So the trypanosomes, they can circulate in the bloodstream, and then they can invade cells within the body. And some of the cells that they can invade include cells of the nervous system, like glial cells. They can invade cells of the reticuloendothelial system. They can invade cells of the heart, so myocardial cells. Now, in some folks might just have maybe a mild fever, a little edema, lymph node swelling, but a lot of people are going to just spontaneously recover. However, one book said approximately a third of the folks may progress to the chronic form. And in the chronic form, we're going to see nerve damage to the gastrointestinal tract. And as a result, we can have a condition called megaesophagus. Megaesophagus. So the esophagus is really big. It's going to be hard to swallow. And then megacolon, where the colon can't contract properly because of nerve damage, and you have feces just piling up in the colon, so you get this huge colon, right? And then another place the trypanosoma like to replicate is in the myocardial cells. So you can end up with a cardiomyopathy, and also let's see, there was a fancy name for this, so I'll have to ask the A&P folks to help me out with this, a right bundle nerve block, so you don't get transmission of the signals required for effective heart beating. So in folks that are chronically infected, they can sometimes die from congestive heart failure. So that's really bad news. And furthermore, when in these chronic cases, when the patient develops megaesophagus, megacolon, or the congestive heart failure, therapy to try to kill the parasite, it won't work anymore because the damage is permanent. There was one book describing the autonomic nerve damage that occurs in some of these chronic cases. They're not sure why. One author proposed that there's a lot of fibrosis associated with infection, and maybe possibly there's some kind of autoimmune disorder that's occurring. Okay, but certainly the parasites can cause harm as they're growing within the cells themselves. So, so more of the ecology here. Trypanosoma Cruzi, Chagas disease, American trypanosomiasis, it is a zoonosis, and sometimes the books don't state that, and I think this is pretty important. So in some studies in South America, in endemic areas, they say a risk factor is if you live with a lot of dogs, because dogs can be infected and act as a reservoir for the reduvid bugs. In addition to dogs, it was listed that rodents can be infected, cats can be infected, lagomorphs such as rabbits, armadillos, opossum, and raccoons can be infected, and so those would be animal reservoirs. Trypanosoma cruzi has been found in donated blood in California, so the blood centers always screen for Trypanosoma cruzi in donated blood. The vector that can transfer Trypanosoma cruzi is present in the southern United States, so there might be some endemic cases, say in Florida, Louisiana, Mississippi. There are possible vectors here in California. The thought is probably a lot of the cases that are diagnosed in the United States, people have gotten infected living in endemic areas, and then when they come to the United States, they maybe develop the chronic symptoms. So not sure on actual endemic transmission here in the United States, but certainly we have the vectors. And if we have people or animals infected, that means that there's a potential for endemic transfer here in the United States. So with regard to diagnosis, you can do the blood smears looking for the little flagellated Trypanosoma cruzi. There are some antibody tests, so you can do serology. Sometimes you can do biopsies to see if you can see the intracellular form of the Trypanosoma cruzi in infected tissues. And then a very kind of fascinating way to diagnose is called Xenodiagnosis. And this is when you take laboratory reared reduvid insects, right, that aren't infected, and you let them take a blood meal on the patient who you think might be infected, and then you raise those reduvids and then sacrifice them, and then look for Trypanosoma Cruzi in their digestive tract. So again, kind of a fascinating way to do the diagnosis, and again, that's called xenodiagnosis. Two of the drugs that they said are kind of classic for treating Chagas disease, I've had a really hard time finding the mechanism. One of the main drugs is called nifurtimox, and it's supposed to be a nitrofuran. So I've been trying to figure out if it might have the same mechanism as the metronidazole. The metronidazole is a nitroimidazole, and so I'm wondering if it's the same mechanism, possibly of activation within the host and then incorporation into the DNA of the parasite. And then another of the drugs, and again, I have to do some more digging, that can be used is called benznidazole. All right. So again, folks, I think we'll stop it here. The next videos will be on Trypanosoma brucei, the cause of African trypanosomiasis or sleeping sickness. And then we'll do which is also arthropod borne. The vector is called glycina, or the tsetse fly. We'll then talk about Plasmodium. Plasmodium causes malaria, and it's transmitted by female Anopheles mosquitoes. And then we'll finish with a bonus on a mutualistic Protozal symbiont of termites called Trichonympha. And it's really cool because Trichonympha make the enzyme cellulase that helps termites digest wood when they eat wood. Okay, so we'll stop this one here, and then we'll be back.